Hello and welcome. This is AQA GCSE Biology, and we're going to look at the whole topic of organisation, which is the second unit for GCSE Biology. Remember, there's timestamps in the description below if you want to skip between topics, and do remember this is an overview revision. It's not in great detail. If you want detailed videos, you can look at the playlist on my channel and you can find all the videos that go along with the topics there. However, let's get going. The first thing we want to look at is principles of organisation and enzymes. So the first thing we need to talk about is the idea of cells. Cells are the basic building blocks of all living organisms. I've just sketched out a quick diagram of a type of cell called a muscle cell, but these are the basic building blocks of living things, and usually they don't work by themselves, they work together in groups, and when we have a group of cells, we call this a tissue. A tissue is a group of cells with similar structure and function. When we have tissues working together, we talk about an organ, and an organ is a group of tissues performing specific functions. So I've gone from muscle cells to muscle tissue to the organ of a muscle. Next we have the organ system. This is a group of organs performing a specific function. When we have organ systems together, we call that the organism. Okay, so these are some key definitions that we need to know and remember for this unit. Now let's move on to enzymes. Enzymes are protein molecules and they are found in cells. Sometimes they work outside of cells, but very often inside of cells as well. Here's two molecules of an enzyme. If we magnify one of those very many times, you might see a structure that can be represented like this. Enzymes are actually a 3D structure, but I've just drawn mine 2D to keep it simple. And an important part of this structure is this part here. This is called the active site. Enzymes are biological catalysts. That means they speed up reactions in the body and they can be reused. Their shape remains unchanged. They depend on their shape to work, and the shape usually, if not in extreme conditions, will stay the same, which means they can be used over and over again. We use a lock and key model to help us explain how enzymes work. Here is a different enzyme molecule. You can see it's different because it's got a different active site. And here is another type of molecule called a substrate. We can describe the substrate as being a bit like a key and the active site being a bit like a lock. The substrate molecule can fit inside the active site. We get a chemical reaction and then we get product molecules. So the substrate is a bit like a key, the active site is a bit like a lock. Each enzyme is specific to one type of substrate molecule. Again, it's just like a key. One key is specific to one lock. So this is why this model is quite useful. The shape of the substrate is complementary to the shape of the active site. We don't say they're the same shape because they're not the same shape. The shapes are complementary. You can also say the substrate molecule fits inside the active site. Enzymes control metabolism in the body. Metabolism reactions are reactions that build up and break down molecules, and these are all controlled by enzymes. We can measure the rate of enzyme-controlled reactions. Sometimes when we have a reaction with an enzyme, a gas is given off. We have the production of a gas, and we can measure that in volume per time. This is usually measured in the unit of centimeters cubed per minute or something similar, but it's got to be a volume and a time. Sometimes there's a color change in an enzyme-controlled reaction, so we can measure the rate of that by looking at the time it takes for the color to change. And sometimes in some reactions we have a change in pH. So again, we can measure the time taken for a certain change in pH. These are just some ways in which enzyme-controlled reactions can be measured. There are factors that affect enzyme action. One of them is temperature. If we look at a graph of the temperature versus the rate of reaction, we see it looks a little bit like this. We can describe this and explain this. So for the first part of the graph, the higher the temperature, the faster the reaction. This is because the enzyme and substrate molecules move faster, they have more energy, they have more kinetic energy, and so they collide more often. The fastest rate of reaction is at the optimum temperature. So on the graph, you can see the optimum temperature is there. And for many living things, the optimum temperature is around about 37 degrees centigrade. At higher temperatures, the enzyme denatures, the shape of the active site is permanently changed, the substrate no longer fits the active site. Here is an enzyme molecule as normal. However, if the temperature is too high, the active site changes shape, the substrate no longer fits the active site, we say that the enzyme is denatured. pH also affects the rate of enzyme-controlled reactions. Enzymes work best at their optimum pH. So if we look at a graph of pH versus rate of reaction, the graph looks a little something like this. There's the optimum part for that reaction, right in the middle there where the graph peaks. When we say they work at their best, we mean they work at their highest rate at optimum pH. At excessive pH, in other words, too high or too low, the enzyme active site can change shape, and again, we say the enzyme can denature at excessive pHs. Different enzymes can have different optimum pHs. So for example, this enzyme here has a lower optimum pH, so works in acid conditions, for example in the stomach. This enzyme has a higher optimum pH, for example enzymes that work in alkaline conditions in the small intestine. So that's organization of cells and tissues and organs in the body and key details about enzymes. Next, we can look at the human digestive system. This also involves enzymes. The human digestive system is an organ system where several organs work together to digest and absorb food. We should be able to label the parts of the digestive system. So we can start at the top there with the mouth. This is the esophagus, also called the food pipe or the gullet. That leads us onto the stomach, and just below the stomach there's an organ called the pancreas. The stomach leads on to the small intestine. Small intestine leads to the large intestine and eventually to the rectum and the anus, where undigested food is removed from the body. We also have the liver, which is there, and just behind the liver there's a structure called the gallbladder, and we'll be looking at the function of those two shortly. In the digestive system, large insoluble food molecules are digested to produce small soluble molecules, and these can be absorbed into the blood. So that's the function of the digestive system. Food is digested by enzymes, and there's some key ones that we need to know about. Carbohydrase enzymes, lipase enzymes, amylase, which is a type of carbohydrase, and protease. I'm going to use these letters here to represent those enzymes. These are not official abbreviations, so you shouldn't use those when you're writing answers to tests. However, we can see that the mouth is a place in the digestive system that produces amylase, sometimes called salivary amylase. The stomach produces protease enzymes and no other enzymes. The pancreas produces all four of them, and the small intestine also produces all four of them. So these are the locations where the enzymes are produced, but we need to know what these enzymes do. So action of enzymes is next. Carbohydrates is a food group that is important in the diet. Here is a diagram of a carbohydrate molecule, or a small part of a carbohydrate molecule. This can be broken down by carbohydrase enzymes, and here is an example of a carbohydrase enzyme. You can see its active site fits that carbohydrate, and it can break that carbohydrate down into simple sugars. We also have an example of a carbohydrate called starch. Here's a small molecule of starch. This is broken down by amylase. Amylase is a different enzyme, so as you can imagine, it has a different active site. The shape of the active site is different, but it works in a similar way in that the active site fits the starch molecule and it breaks it down also into simple sugars. Remember, amylase is a type of carbohydrase enzyme. Once we've broken these down into the simple sugars, they can be used in the body. For example, a simple sugar is glucose, which can be used in respiration, or the simple sugars can be built into new carbohydrates, for example, glycogen, something we'll look at in a future video. We have proteins, and proteins are made of long chains of these units called amino acids, and proteins are broken down by protease enzymes into those amino acids. We have lipids. Lipids, also known as fats, are broken down by lipase enzymes into something called glycerol and fatty acids. glycerol, and fatty acids. And these amino acids and fatty acids, these can be built into new proteins and new lipids that the body needs. We need a little bit more detail about the digestion of lipids, so let's take a look at that. The digestion of lipids. We just need to look at this part of the digestive system. We have the liver, as we talked about before, but the liver produces bile, and this structure called the gallbladder, this stores bile. Bile passes into the small intestine to help digest lipids. Two things. One, bile is alkaline, so that neutralizes stomach acid, allows the lipase enzymes to work better in the small intestine. And two, bile also emulsifies fats. In other words, it makes large droplets or large drops into small droplets. Let's have a look at that a bit more visually. These are our large lipid drops. The surface area, the total surface area of this is quite low. We could say it has a low surface area to volume ratio. After the bile is added, we get emulsification and we end up with small lipid droplets. These have a much higher surface area or a higher surface area to volume ratio. So lipase enzymes work best in alkaline conditions, and the high surface area increases the rate of the action of lipase. Remember, bile does not digest the lipid, it just emulsifies it and makes it easier for the action of lipase. Lipase is the enzyme that does the digesting. Next, we're going to take a look at food tests. The first one is the iodine test for starch. Here is some bread. Here is some iodine, which is an orange color. It's called iodine solution. We add the iodine solution to our sample, in this case the bread, and if we get a blue-black color, we know starch is present. So the test is add iodine solution, which is orange. If the color changes from orange to blue-black, we have the presence of starch, or starch is present. That's the first food test we need to know. We can use other food samples as well. Sometimes you need to crush them up and maybe add a bit of water, but potato, rice, pasta, all that kind of stuff works well with the iodine test for starch. Next, we have the biuret test for protein. Here's a sample. Again, it's either crushed or blended, or it might be liquid like milk, something like that. And here is our biuret solution. This is blue, and we can add it to our sample, usually an equal volume compared to the sample. We add our biuret solution. If we get a lilac color, we know we have the presence of protein. So that's our test for protein. The next one is the test for lipids. Here we have a sample again, crushed or blended if we need to. We add a few drops of ethanol. We then shake, so you might have to put a rubber bung on the top and give it a shake. And the test is to add ethanol, shake. We then add some water, and if we see a cloudy white emulsion, this indicates lipid is present. If it goes cloudy white, we know we have lipid. And that would be something like that. It's a kind of grey colour on my diagram there, but we mean a cloudy white emulsion. The next test is the Benedict's test for sugars. Here is a sample, again crushed or blended. Here is our Benedict's test, a very similar colour to our biuret solution, our Benedict's reagent, a very similar colour to our biuret solution. We add that to our sample. We then have a slightly different procedure. We have to heat this in a water bath. So we heat it in a water bath for a few minutes and we see a color change. So the test is we add Benedict's reagent, we heat in a water bath. The color change is from blue to brick red, and this indicates the presence of sugars. You do actually see a range of colors that form. So it goes from green to yellow to orange to a brick red color. Here is a summary of all those tests, and you can look at that in your own time and make sure you are clear with those. Next, we're looking at the heart and blood vessels. Lots on the next slide, so take a deep breath and here we go. The heart and circulation. This is the circulation system in a very simplified diagram. Firstly, on one side we have oxygenated blood going through the blood vessels, and on the other side we have deoxygenated blood. This is the heart. This is the lungs, and this indicates the whole body, including the head. This is the right side and this is the left side. We switch it around because we do it from our own perspective rather than the perspective of it being on the page. So we switch left and right around. This major blood vessel coming out of the heart is called the aorta, and this carries oxygenated blood to the body. Once the blood gets to the body, we have some gas exchange, and then the blood goes back to the heart and enters it via the vena cava. This carries deoxygenated blood from the body. This is the pulmonary artery. It goes from the heart to the lungs, carries deoxygenated blood to the lungs. In the lungs we have gas exchange, so carbon dioxide is removed from the blood and oxygen is taken in from the blood from fresh air. And then the blood travels down the pulmonary vein back to the heart again. It carries oxygenated blood from the lungs back to the heart. Another point to mention is that oxygenated and deoxygenated blood is kept separate in the heart. There's some tissue in the middle of the heart that keeps the oxygenated and deoxygenated blood separate. The other thing to mention, if we just magnify that image a little bit, is we have a set of valves that you can see there. There's 4 valves that have been highlighted. And the valves, they stop the backflow of blood. So the way they do that, so here is some valves there in the darker color, the blood in the red arrow. They open, they allow the blood through, but they will not allow the blood to go backwards. The other valves going out of the heart, they do the same thing. The blood can travel in one direction only. The blood cannot travel backwards for properly functioning valves. We call this system a double circulatory system. This is where blood is pumped to the body by the left side of the heart and to the lungs by the right side of the heart. That's one way of explaining what a double circulatory system is. We could also say that in one full circuit around the body, blood enters the heart twice. A quick mention here that the lungs are involved in transferring oxygen around the body in that they take in oxygen from the air. We need to know a couple of labels on this. This is the trachea, which is the windpipe. The windpipe branches into 2 smaller tubes called the bronchi. If you've got 1, it's called a bronchus, 2 is called bronchi. And then at the end of the bronchi, we have smaller vessels or smaller tubes eventually leading to a structure called an alveolus. We've talked in detail about the structure and function of that in the previous video, which you can go back and watch when you get a moment. Let's label the parts of the heart. This is called the left atrium. Remember, left and right is switched when you're looking at it on the page. So this is the left atrium. This is the left ventricle. Remember, the left ventricle has thicker walls than the right ventricle because it has to pump blood further than the right ventricle. This is the right ventricle, and this is the right atrium. The valves are here as shown, and as we saw previously in the previous diagram, and as we know, they stop the backward flow of blood. In the right atrium, in the corner there's a small structure called the pacemaker. This controls the resting heart rate. It makes the heart beat periodically. It sends regular impulses that makes the heart beat regular. Sometimes the heartbeat is not regular. You can see here this person is fitted with a device, and that's called a pacemaker. It's an artificial pacemaker, and it helps to send regular impulses across the heart for a heart that is not beating properly if the heartbeat is irregular. It's an electrical device that sends regular impulses to the heart and helps regulate the heartbeat. Next, we need to look at the structure of the blood vessels. So let's start with arteries. Here is an example of an artery. It's what we call a cross section. So if you've sliced it in half crossways, then that's what it would look like. We have the part inside where the blood is carried. That's called a lumen, and arteries have a narrow lumen. There's a thick layer of muscle around the artery, and this can contract to pump blood. And we also have a thick elastic tissue layer as well. This recoils, in other words, springs back, and that maintains the pressure in the artery to keep the blood flowing. The thick muscle and the thick elastic tissue, they also withstand the high pressure that is found in arteries. Next, we're going to go to the smallest blood vessels. These are capillaries. So here is a cross section of a capillary. If I was to draw it to the right size, the right scale, we wouldn't be able to see it because it's absolutely tiny in comparison. But we've enlarged it so we can see the parts. And The first thing is that it has very thin walls. The thin walls are only 1 cell thick, so that makes them very thin. This allows for a short diffusion distance for gas exchange, where the oxygen is leaving, going into cells, or coming in from the lungs. The diffusion distance is very short, so diffusion happens quickly. Capillaries are very narrow. That means red blood cells have to travel in a single line. Red blood cells carry oxygen, so again they are very close to the walls of the capillary, which means the diffusion happens much more rapidly because again a short diffusion distance. There are many capillaries in the body, and that gives a large surface area for gas exchange. Next, we're going to take a look at veins. So let's look at a diagram of a cross-section of a vein. It's a bit more like that, a bit flatter and less round. The lumen is much larger in veins compared to arteries. The walls are made of thin elastic and muscle tissue, and that's because veins have much lower pressure in them, so they don't need to have thick walls. And they have valves. Arteries most of the time don't have valves. Veins very commonly do have valves. They stop the backflow of blood, as we said before, and that means blood will only move forwards, or it will only move towards the heart. It won't move away from the heart. The valves stop the blood from flowing backwards. So here is blood being able to flow forwards in the left-hand diagram. If it tries to flow backwards, it cannot because the valves close. So that's the heart and circulation and blood vessels. Next, we're going to take a look at the blood and coronary heart disease. Let's have a look at the components of the blood. Here is a small sample of blood. If we were to magnify that, we would see that it has different parts in it. This part here, the liquid part, is called plasma. We have a white blood cell. We have a red blood cell there, lots of red blood cells in that image. And we have these little structures called platelets. White blood cells have the job to kill or destroy harmful microbes, otherwise known as pathogens. Pathogens are microbes that cause disease. The plasma, this is the liquid part of the blood, and it's mostly water. However, it carries all the red blood cells, all the white blood cells, and it transports dissolved substances, for example glucose or amino acid or even hormones that need to be transported around the body, and glucose and amino acids being the products of digestion that we talked about earlier. Red blood cells, they contain lots of hemoglobin, which carries oxygen. If you look at a cross-section of a red blood cell, one, you can see it has no nucleus, so there's more space to carry oxygen, and it can carry a lot of hemoglobin, and it has a shape that we call a biconcave shape, so it's kind of caved in on both sides. That increases the surface area, which allows the red blood cells to absorb more oxygen and release more oxygen because of that high surface area. We then have these little structures called platelets. These are small cell fragments which help blood to clot. Clots can block wounds to stop bleeding, so they are very important to stop blood loss in case we get injured. Finally, important to remember that blood is called a tissue, or is thought of as a tissue, and it's made of plasma in which red and white blood cells and platelets are suspended. Let's have a look at coronary heart disease as the next part of this unit. Here is the heart. Here is the major blood vessel called the aorta, which, if you remember, carries blood from the heart to the body, and it carries oxygenated blood. However, that's not the blood vessel we're looking at. We're looking at these blood vessels in that dark red color, and these are the coronary arteries. These supply the heart muscle with oxygen and glucose. Sometimes these can get blocked. This is a cross-section of a coronary artery. Sometimes fatty material can narrow these arteries. That fatty material comes from a poor diet. So there you can see that blockage in that kind of yellow color. That means less blood is going to be able to flow through that artery. Less blood flow means less oxygen to the heart muscle. That means less aerobic respiration, therefore less energy release for muscle contraction or heart muscle contraction. And that, of course, can be very dangerous and could lead to a heart attack. Next, we need to look at the possible treatment for coronary heart disease. The first treatment is using drugs called statins, and these are drugs that slow down the buildup of fatty material in arteries. There are sometimes side effects, including kidney failure or muscle damage in some patients. So there are possible negative effects of taking statins. We can use something called a stent. This involves surgery. So here is a side view of a blocked artery. This is a narrowed artery. A stent is a little device that's by surgery placed into the blood vessel, the artery, made to expand using a balloon, and that will then hold the artery open. So before you could see it was very narrow, now it's widened and more blood can flow. A stent is used to keep arteries open to improve blood flow. However, there is a risk of infection or damage to blood vessels from surgery, or a risk of clots. So this carries risk as well. Another issue that can happen in the heart is faulty heart valves. So here is the heart, and here are two of the valves. Sometimes the valves don't open fully. So if you look at the one on the left, it's opened partially. The blood can only partially pass through, so that reduces the amount of blood passing through those valves. Or sometimes we have an issue where the heart valve is leaky. Therefore, when it closes, it doesn't close completely, and some blood can pass in the backward direction. This is a problem because it can reduce blood flow to the body. Not enough oxygen gets to the body cells. That means less aerobic respiration, so less energy release for muscle contraction. Less respiration, so less energy release. This can lead to tiredness, difficulty exercising, or even chest pain because the heart has to work much harder to pump the blood. Faulty heart valves can be replaced. The replacements can be either mechanical or biological. A mechanical heart valve is made of metal or plastic polymers. These are long-lasting. There's less or no surgery needed in the future, or much longer time passes before surgery might be needed again. There is, however, a risk of clotting, so patients may have to take drugs called anticoagulant drugs, and these stop the blood or reduce the risk of the blood from clotting. We can have biological valves. These come usually from a pig or a cow. They may need replacing after 5 to 10 years. That should say 5, not 50. There you go, that's fixed. We do have, however, a risk of rejection. We do have the risk of rejection, the body rejecting the valves from a pig or a cow, so the patient may need to take immunosuppressant drugs for the rest of their lives. So the patient may need to take immunosuppressant drugs. These are drugs that weaken the immune system, so the person is less likely to reject the valves from a pig or a cow. Next, we're going to take a look at heart disease and health issues. Sometimes issues can be so severe that it leads to heart failure. Heart failure is when the heart stops pumping for a variety of different reasons. If it fails completely, there is a possibility of obtaining a donor heart or heart and lungs, and these can be transplanted. We can even, in some cases, use artificial hearts, and these are there to keep patients alive while waiting for a transplant. Sometimes they're used for a short while the heart recovers after some damage or after a heart attack. However, all surgeries carry risks, for example, infection or tissue or organ rejection. Next, we're going to look at the issue of health in other areas. Health is the state of physical and mental well-being. Poor health is caused both by communicable and non-communicable diseases. Communicable diseases are ones that can be caught. They are caused by pathogens. Non-communicable ones cannot be caught. These are ones like heart disease or cancer or that kind of thing. Diet, stress, and life situations may have a big effect on both physical and mental health as well. Different diseases can interact. This is a white blood cell. If there is a defect in the immune system where white blood cells do the job of protecting against pathogens, this can lead to an increase in the chance of suffering from infectious diseases. If the white blood cells are not working well, there's a higher chance that the body can suffer from infections. Viruses, these reproduce inside body cells, and they can sometimes trigger some sorts of cancers. Also, severe physical ill health can lead to depression and other mental illnesses, and in addition, immune reactions caused by a pathogen can sometimes trigger allergies, for example, skin rashes or asthma. The next thing is to do with cancer. Cancer can arise as a result of uncontrolled cell division. That will lead to tumours. This is a malignant tumour. This is a benign tumour. The malignant tumour is the one that is cancerous. This can invade neighbouring tissue, it can spread in the body and form what's called secondary tumours. So it can form different tumours around the body. For a benign tumour, these are abnormal cells, but they are all contained in one area. We don't describe these as cancerous, and they are often surrounded by a membrane, which makes them quite easy or easier to remove with surgery, depending on where they are in the body. Next, we need to take a brief look at the effect of lifestyle on non-communicable disease. There are risk factors linked to an increased rate of disease, and risk factors can be aspects of a lifestyle, or they can be substances in the body or the environment. Some risk factors have causal mechanisms. So, diet, smoking, and lack of exercise is a risk factor for cardiovascular disease. Obesity, that means being severely overweight, is a risk factor for type 2 diabetes. Overuse of alcohol is a risk factor for liver and brain disease or liver and brain poor functioning. Smoking is a risk factor for lung disease and lung cancer. Smoking and alcohol is linked to poor health of unborn babies. And substances called carcinogens, these are chemicals, and these can lead to cancer. Carcinogens are often found in cigarette smoke. Here is an example of something you should be able to do. This graph is a scatter graph. It shows a link between body mass and the number of people with type 2 diabetes. It shows a positive correlation. The higher the body mass, the higher the number with type 2 diabetes. However, you have to be careful with a graph like this. It shows a positive correlation, but it doesn't prove a link. It doesn't prove that a high body mass is going to give type 2 diabetes, but it does show a positive correlation. Plus, we need a lot more data. There's only a handful of people that have been sampled in this. We don't know the age. We don't know whether male or female. We don't know various things in this population. So we have to be careful with data like this. If you've made it this far, you're doing really well. Not long to go now. We're going to look at plant tissues and organ systems in plants. Plant tissues. Let's take a look at a leaf. This is a leaf. Part of it has been sliced off. A leaf is a plant organ with different tissues in it. If we magnify that region there and look at it under a microscope, you might see a structure that looks a bit like this, and we have to be able to describe the different parts of the leaf as shown in the diagram, in the second bigger diagram. We have something called a waxy cuticle. It's a layer of waxy material that's on the top surface, usually on the top surface of leaves. This is transparent, waterproof, and it reduces water loss from evaporation. Water is very important to a plant, and we don't want to lose it in an uncontrolled way. This is called the upper epidermis. This is a transparent and protective layer. There are no chloroplasts in that layer. The next layer is called the palisade mesophyll layer. Remember, palisade mesophyll is a tissue made of palisade cells. This contains many chloroplasts to absorb light for photosynthesis, and it kind of makes sense because it's on the top surface of the leaf where most of the sunlight will fall. This layer here is called the spongy mesophyll. It contains some chloroplasts, but it has air spaces between the cells to allow for diffusion of gases. This is the lower epidermis. This has a structure called a stoma. If you've got many of them, they are stomata. And stomata are little openings in the bottom of leaves, usually in the bottom of leaves, and they are made up of two guard cells. Stomata can open and close to change the amount of gas exchange and to control water loss. This is xylem tissue. This will transport water into the leaf, and below that is what's called phloem tissue that transports sugars out of the leaf. One more detail about stomata. Here is a top-down view of a stoma, and it's closed. These are the two guard cells. We have a thin cell wall on the outside part, and we have a thicker cell wall on the inside part. That means that to open, water can be absorbed by the guard cells by osmosis, and that causes the stomata to open. The guard cells swell and the stoma opens. Transport in plants. Here is a plant. Dissolved sugars will go from leaves to the stem. This is the phloem that's found in the stem. We've talked about this in the previous video. It then goes potentially to the roots and to storage tissues. This is called translocation. One thing to remember, as shown by the blue arrow, is that it's the movement of dissolved sugars and other molecules around the plant, both up and down the plant, not just in one direction. Water enters through the roots, goes up the stem, into the leaves, and evaporates out from the leaves. So water goes into root hair cells, it goes into the roots, the stem, and in the stem we have the xylem. We actually have xylem in the roots as well as the stem, but this carries water and minerals up the plant, into the leaves, and then it evaporates out through the stomata. This is called the transpiration stream, the movement of water up the plant. Transpiration, however, be very careful with this definition, it's slightly different. It is the evaporation of water from the leaves through the stomata. So transpiration is just the evaporation of water through the leaves, whereas the transpiration stream is the movement of water all the way through the root hair cells, all the way up through the plant, and eventually out through the stomata. There are different factors that can affect the rate of transpiration. Temperature, air movements, by air movement we mean wind, light intensity, and humidity. I'm going to use an upward arrow for the word higher or more, and a downward arrow for lower and less. So a higher temperature means a high rate of transpiration. This is because water particles have more kinetic energy when they're at a higher temperature, and so they are more likely to escape, and therefore the water evaporates more rapidly. High wind means more transpiration. That's because water particles are moved away from the surface of the leaf, and the water concentration in the air is kept very low. So there's going to be more diffusion of water out, in other words, more transpiration. High light intensity means a high rate of transpiration, and that is because there are going to be more stomata open, and as water evaporates out of the stomata, more water is drawn up, and this is for photosynthesis. And it makes sense. If you have a high light intensity, you want to take advantage of that to increase the amount of photosynthesis. High humidity means a lower rate of transpiration because water concentration is high in the air, and that means that there is going to be a less chance of water evaporating. So high humidity reduces the amount of evaporation from the leaf, reduces the amount of transpiration. So there we have it. That was the whole of the organisation topic for AQA GCSE Biology. Thank you for watching, and I'll see you soon.